At 14,000 feet, the human body begins to betray itself. The process is gradual and at first deceptive. The lungs pull in air at the same rate they always have, the same number of breaths per minute, the same reliable rhythm that operates below the threshold of awareness. But here, on the high plateau of Tibet, that air carries roughly 60% of the oxygen available at sea level. The body notices. Deep in the bone marrow, in the molecular signaling that runs beneath any conscious experience, a response triggers. Make more hemoglobin. Produce more red blood cells. Carry more oxygen per cubic centimeter of blood to compensate for the shortage in every breath. This works. For a while, but blood thick with extra red cells becomes sluggish. It struggles through capillaries with increasing resistance, strains the heart's right ventricle, thins the margins of the brain's oxygen supply even as it tries to improve them. In the Andes, where different populations have spent their own millennia at altitude, a condition called chronic mountain sickness claims a significant portion of highland men as they age. The face acquires a permanent deep flush. The fingers swell. The heart, overworked for years, eventually enlarges and fails. This is what the human body does at high altitude. Most human bodies. On the Tibetan Plateau, something different happens. The hemoglobin levels of Tibetan Highlanders, measured, confirmed, and reconfirmed across decades of clinical research, do not spike upward in the presence of altitude. In many groups, they run lower than sea level norms. The blood stays thin. The heart stays unburdened. And yet the cells are getting oxygen, extracting what they need from air that would leave an unacclimatized lowlander confused and gasping within hours. Children grow up at 15,000 feet and run. Old people walk the plateau roads in winter wind without stopping. Physiologists documented this for decades. It was real, reproducible, biologically extraordinary. But where it came from was a different question entirely. Natural selection can reshape a population, but it takes time. Modern Homo sapiens have been on the Tibetan Plateau for perhaps 30,000 years, a significant span, but researchers looking at the specific gene variant responsible for this difference found it too precise, too thoroughly dominant to be the product of that time frame alone. The variant sits in a gene called EPS1, which governs the body's response to low oxygen. It is vanishingly rare in every other human population on Earth. It didn't drift in slowly from some ancient shared ancestor. It arrived in Tibetans from somewhere else. From someone else. 40 years ago, no one could have said who. 10 years ago, the answer had just become clear. The EPS1 variant in Tibetan genomes is most similar, genetically, to the genome of a people who were, at the time of that discovery, known from a single piece of bone. This is the story of that bone, and of the people it belonged to. The entire physical evidence for the Denisovans, every fragment, every chip, every object we can hold in a gloved hand and say with confidence belongs to them, would fit without crowding on a single museum shelf. A distal phalanx, a fragment of finger bone, recovered in pieces, 3 oversized molars, each one substantially larger than its Neanderthal or modern human equivalent. A thin sliver of parietal bone from the skull, smaller than a matchbook, and a lower jaw found in a cave on the Tibetan Plateau, 600 miles from where everything else was discovered. That is the Denisovans. That is the total skeleton of a species. By any normal standard of paleontology, this should tell us almost nothing. Scientists have reconstructed extinct creatures from less. It's true, but usually with comparative anatomy to guide them, related forms to provide context, a surrounding ecosystem to place the discovery within. The Denisovans offer almost none of that. No confirmed stone tools. No confirmed hearts we can point to as exclusively theirs. No art, no burial, no ornamentation we can say with certainty. This is what they made. A people who appear to have endured for 400,000 years or more across one of the largest territories of any hominin ever identified. And they left behind less physical matter than a single individual buried in a Neanderthal grave in a rock. And yet, we have their genome. Not a fragment of it. Not a degraded smear of sequence painstakingly reassembled from contaminated sediment. From a single individual, researchers produced a high-quality, high-coverage genome. One that, in some technical respects, rivals what a modern laboratory would generate today from a fresh blood sample. From that genome, and the cascade of analysis that followed over the next decade and more, we know how the Denisovans were related to Neanderthals. We know something about what they probably looked like. We know that modern humans and Denisovans met and had children together, not once, but at least twice, in at least 2 different parts of Asia, and that the descendants of those children are alive today, carrying Denisovan DNA in their cells, some of it still performing specific biological functions. The Denisovans are people we know from the inside out. We have read their blood before we found their bones. This documentary is the story of how that became possible. It is a story about a scientific revolution built by people who spent decades solving problems that had never been solved before. It is a story about what the ancient world actually looked like in the long centuries before our own species arrived in Asian force. How many kinds of human beings shared the earth, how they encountered one another, what passed between them. And it is a story that is not finished. The field is alive and active. Discoveries are arriving regularly. Some of what we understand today will be revised or overturned within years. Stay with us. By the time this is done, you'll understand why the people of Tibet can breathe where others cannot, and where that ability came from. You'll meet a teenager who was, in the most precise biological sense, half one species and half another, and you'll understand what her existence reveals about what ancient human life was really like. You'll understand why geneticists working in Germany found the traces of an unknown population living in Siberia, encoded in the genomes of people in Papua New Guinea. And you'll sit with a question the science opens but doesn't resolve. If a people this widespread, this durable, this genetically significant can leave almost no physical trace, what else are we failing to see? To understand the Denisovans, you have to begin in a world that has no surviving name, no language to describe it, no record of what the people living in it call themselves, or each other, or the land they moved across. We are somewhere between 600,000 and 750,000 years ago. The precise figure is contested. The genetic models that estimate divergence times depend on assumptions about mutation rates that are themselves revised with each new dataset. But the broad shape of the story is not in dispute. Africa is the center. It has been the center of hominin evolution for millions of years and will remain so for a long time yet. But the genus Homo is not confined to Africa by this point. Homo erectus, upright, long-striding, with a brain substantially larger than any earlier hominin, had pushed out of Africa well over 1 million years before the period we're entering now. Their descendants, or perhaps parallel branches of the same spreading lineage, are present across an enormous stretch of the Old World. The islands of Java, the cave sites of northern China, the wide plains of the Indian subcontinent. They've been in Asia long enough to have adapted regionally, developed local characteristics, become, in some meaningful sense, Asian people. In Africa, meanwhile, and possibly in parts of Eurasia that maintained intermittent contact with Africa, a new type is emerging, larger brained than Homo erectus, more sophisticated in its stone tool technology, physically powerful with heavy brow ridges and robust faces, but with a brain case that is expanding towards something new. Paleontologists apply the name Homo heidelbergensis to many of the fossils from this period, though the classification is contested. The specimens come from places as far apart as Zambia, Ethiopia, Greece, and France, and they are varied enough that some researchers think the category covers too much ground to refer to any single biological species. What none of them dispute is a general pattern. Somewhere in this period, the lineage that will eventually produce modern Homo sapiens begins to diverge, definitively and permanently, from the lineage spreading through Eurasia. The split isn't a single event. It is a process. Populations losing contact, gene flow slowing, then stopping, mutations accumulating differently on either side of the gap that is widening generation by generation into something uncrossable. Out of that divergence, 2 major lineages eventually emerge in Eurasia. One moves west into Europe and Western Asia. The other moves east. The western branch, the Neanderthals, is the one we know. They lived for hundreds of thousands of years across Europe and into Western Asia. They buried their dead. They crafted jewelry from eagle talons and perforated shells. They made stone tools of real technical sophistication, managing the pressure and angle of every flint strike to produce specific cutting edges. Dozens of Neanderthal skeletons have been found, analyzed, and argued over. We have sequenced multiple Neanderthal genomes. Whatever we once thought about Neanderthals being primitive or brutish has been substantially dismantled by the evidence of the last 3 decades. They were people, in every meaningful sense of the word. The eastern branch is different. The eastern branch is almost invisible. They move deeper into Asia, past the Caucasus, past the great steppe belt that runs from Eastern Europe towards Central Asia, into the enormous interior of the continent, into territories where, even today, the human presence is thin. Vast grasslands, folded mountain chains, river systems that drain into closed basins with no outlet to the sea. The winters here are extreme. The terrain is always demanding. And the fossil record is, for reasons both geological and archaeological, exceptionally sparse. This eastern lineage, what we now call the Denisovans, existed for hundreds of thousands of years across this territory. The genetic record, recovered from those handful of bones, implies a population that was once large and geographically widespread, not a small refugium population clinging to a single valley. Something extensive. The diversity visible in the genomes of the few individuals we've sampled suggests there were many Denisovans once, spread across many regions, living under many different conditions. How extensive? This is one of the genuinely unsettling things that genetics reveals. The Denisovan signal in modern human populations doesn't cluster around Siberia, where all the physical evidence has been found. It clusters in Southeast Asia, in New Guinea, in Aboriginal Australian populations, in the islands of Pacific. The populations with the highest Denisovan ancestry living today, 4 to 6% in some Melanesian groups, live as far from the Altai Mountains as it is possible to live while remaining in Asia. Whatever the Denisovans were, they were not a local Siberian curiosity. They were something continental, something that extended, in ways we're still working to understand, across the full width of the world's largest landmass. The Pleistocene world they moved through was not the world we know. During the cold peaks of the great glacial cycles, which recurred roughly every 100,000 years, driven by subtle shifts in the Earth's orbit, so much water was locked into ice that sea levels fell by 100 meters or more. The Indonesian archipelago, those thousands of modern islands, was during cold periods largely a peninsula of the Asian continent. A person walking south from the mainland would have passed over land that is now the floor of the Java Sea. The shallow Sunda Shelf, encompassing most of the seas between mainland Southeast Asia and the islands beyond, lay above water during glacial maxima, a territory of rivers and forests and grassland that exists now only as bathymetric contour lines on oceanographic charts. The Denisovans knew that world. The genetic signal suggests they spread into it, but their bones are in Siberia. This contradiction, genes in the tropics, bones in the cold north, is one of the central puzzles of Denisovan research, and we will return to it more than once. For now, it is enough to know that the physical record and the genetic record are telling different parts of the same story, and that neither one alone is the whole picture. There is also the question of who the Denisovans were, biologically, before they became themselves. Their ancestor in Asia, the population they descended from, is not clearly identified. Some researchers believe they descended from an Asian Homo erectus lineage, or a hybrid of that lineage with groups that had migrated in from the West. Others think the picture is more tangled still, involving multiple populations coming together and diverging across an enormous time span. The honest answer is that we don't know. The Asian fossil record from the mid-Pleistocene is sparse, partially excavated, and still being reinterpreted. What we can say is that by the time the Denisovans are clearly detectable in the genetic record, they are already a distinct lineage, related to Neanderthals more closely than to modern humans, but different from Neanderthals in ways that reflect hundreds of thousands of years of separate evolution in very different environments. What we can also say is that they were not alone. The mid-Pleistocene world was crowded by the standards of hominin history. Homo erectus or its descendants were still present in parts of Asia during periods when the Denisovans were also there. Neanderthals occasionally pushed east beyond their European heartland. And then, at some point, the genetic models estimate between 60,000 and 200,000 years ago. The range is wide because the models are imperfect, Modern Homo sapiens begins moving out of Africa in earnest. The world was not empty when we arrived, but we are getting ahead of ourselves. Before any of that, before the meetings and the mixing and the traces written in living DNA, before the laboratories and the Nobel Prizes and the discoveries that rewrote the story of what we are, there is just Altai, a mountain range in what is now southern Russia, running roughly 2,000 kilometers from north to south, rising to peaks of over 4,000 meters. In winter, the temperatures fall past -30. The rivers freeze hard. The forests of pine and larch stand in silence under snow that can last 7 months. In a limestone valley somewhere in those mountains, beside a river that still runs cold and clear, a cave sits in a low cliff face, manageable, warm enough by the standards of Siberia to be habitable year-round, cool enough, as it turns out, to preserve organic material buried in its floor for tens of thousands of years without it fully decomposing. A geological accident, and a fortunate one. Something was living there for a very long time, perhaps with long interruptions for hundreds of thousands of years, sitting at the cave entrance, watching the valley below, making whatever choices a people makes across the long, ordinary years that leave no mark on any record. We just don't know their name yet. Drive south from Bisk in the Altai Republic of Russia, a different political entity from the Altai Krai to its north, and a different entity still from the Republic of Kazakhstan just to the south. Distinctions that matter in the administrative geography of western Siberia and are easy to lose from a distance. And after about 150 kilometers, the landscape changes its character. The road descends into the valley of the Anui River. Limestone bluffs rise from forested slopes on both sides, and the river runs green and fast over stones. In late summer, when the excavation teams arrive and set up camp, the valley smells of wild thyme and pine resin. In November, it smells of nothing except cold. The cave sits in a low bluff on the right bank of the Anui, not far from the village of Chorny Anui. From outside, it is not dramatic. No soaring arch, no cascade of stalactites visible from the road. It is a wide, shallow opening in pale limestone, roughly 20 meters across at the base, giving onto 3 interior chambers arranged in an irregular pattern, a main gallery that is the largest of them, and 2 smaller galleries opening to the east and south. The floor is sediment, ash and bone and pollen, and the stone fragments of tools, the compressed and layered residue of everything that lived or died or was carried inside across hundreds of thousands of years. The ceiling is high enough to stand beneath without difficulty. The walls near the old hearth sites carry the dark stain of ancient fires. What makes the cave remarkable, in the context of ancient DNA research, is something you cannot see or smell or touch. Its temperature. Denisova cave maintains an average of approximately 0 degrees Celsius throughout the year. Not quite freezing. Not quite above it. A stable liminal cold that radically slows the degradation of organic material buried in the sediment floor. DNA, which under most conditions fragments and becomes unreadable within centuries, can survive here for tens of thousands of years, in some layers, as researchers would eventually discover, for far longer. The cave is, in effect, a natural archive that nobody designed and nobody knew was running. The cave has a name. The name has a story. In the 18th century, a Russian Orthodox hermit known as Denis, Dionysi in the formal rendering, withdrew from the world and took up residence here, in what was in a remote corner of the Russian imperial frontier. He was one of a tradition of religious recluses who sought isolation in difficult terrain, and this valley suited him. Cold, quiet, a day's hard travel from anything that could be called a town. He lived in the cave for some years, died somewhere in the surrounding landscape, and passed out any surviving record. But the cave kept his name forward into modernity. Denisova Pestra, Denisova Cave, a hermit's retreat, eventually renamed in the 21st century for a species of archaic human he never knew existed. Denis never knew what was under his feet. Scientific attention reached the cave in the 1970s, when the Novosibirsk researcher Nikolai Ovidov conducted preliminary excavations and found traces of ancient human occupation. Animal bones with cut marks, stone tools, charcoal residue from old fires. Interesting, but not yet transformative. What followed was more sustained. Beginning in the 1980s and continuing without interruption to the present, The Institute of Archaeology and Ethnography is the Siberian branch of the Russian Academy of Sciences, based in Novosibirsk and directed for much of this period by Anatoly Derevienko, undertook what became one of the most methodical archaeological programs in the history of Siberian fieldwork. Derevienko's subject was the Middle Paleolithic of Central and East Asia, the long period, roughly 50 to 300,000 years ago, when hominins were present across the continent, but before modern humans arrived in force. His question was not dramatically different from the questions driving paleoanthropologists elsewhere in the world. Who was here? When? And doing what? But the Altai region offered something unusual. A dense concentration of cave sites and limestone terrain with good preservation conditions, allowing comparative analysis across a regional landscape rather than reliance on isolated finds. In the surrounding valleys, his teams were working multiple sites simultaneously. Okladnikov Cave, about 100 kilometers from Denisova, had already produced what were identified as Neanderthal remains. Shagarskaya Cave, further to the west, would later also yield Neanderthals, individuals, as subsequent genetic work would show, related to populations from Eastern Europe, suggesting movement across considerable distance. The Altai was not a backwater. It was a place where populations converged. Denisova Cave was the richest site of the group. The sediment record goes deep. The lower levels, those furthest from the present surface, contain stone tools and animal bone that preliminary dating places at 300,000 years or more, though precise dating at those depths remains technically demanding, and the numbers carry significant uncertainty. Moving upward through stratigraphy is literally moving through time, each layer marking a different phase of occupation, differentiated by shifts in climate signals preserved in the pollen record, in the character of the animal communities, in the style and technique of the tools. The fauna alone spans an almost incomprehensible range of time. Cave bear remains appear in enormous quantities throughout the record. Bears use the cave as a hibernation site across many periods, and their accumulated bones form a background presence in nearly every level. But threaded through bear bones are the remains of prey species, woolly rhinoceros, cave hyena, ancient horses, ibex, bison, red deer, species now extinct or pushed to the margins of their former ranges, represented here by fragment of bone, carrying the V-shaped cut marks left by stone tools. Someone stripping hide, removing muscle, cracking long bones from marrow. Ordinary work, repeated across generations and millennia, and preserved by cold and limestone until it could be read 60,000 years later. The tools are not crude. This matters because there was for a long time an implicit assumption, present in much of the literature, without being quite stated, that the hominins of Siberia during this period must have been technologically behind their European counterparts. The evidence at Denisova challenged that. The assemblages from the relevant layers include tools made by Levallois technique, a method requiring careful pre-shaping of a stone core to allow the removal of a specifically intended flake, which demands both technical knowledge and planning for a result that doesn't yet exist. A sophisticated cognitive move, and not an isolated one in this cave. And then there was the bracelet. From layer 11, one of the layers that would later become central to the Denisovan story, excavators recovered fragments of a small, curved object carved from chlorite, a dark greenish stone. Cleaned and assembled, the pieces resolved into something without precedent in the regional archaeological record for that period, a polished ornamental bracelet. Its surface had been worked to a smoothness that requires hours of patient abrasive labor, grit against stone, progressively finer, in the same basic sequence a lapidary uses today. It had been drilled through a circular hole, apparently by a rotary technique, which is not widely attested in Middle Paleolithic contexts. Its curvature was uniform throughout, suggesting either precise measurement or an eye capable of maintaining a consistent radius through the entire shaping process. Whoever made this object was not making it to survive. A polished stone bracelet has no caloric value, no cutting edge, no practical function beyond what it says about the person wearing it. It is decoration. It is self-expression. It is a physical residue of a mind that thought about appearance, about how to present itself to others, about beauty in some form. The attribution of the bracelet to Denisovans specifically is contested, Layer 11's deposition overlaps with a period when modern humans were beginning to enter the Altai region, and some researchers have argued that the object's sophistication makes Homo sapiens the more likely maker. Others counter that the layer's dating favors an earlier occupant and that assigning technical complexity exclusively to modern humans assumes the very conclusion at issue. The debate remains open, but it points towards something that will come up repeatedly in this story. Whoever was in this cave, they were not operating at the margins of what we mean by human. By the summer of 2008, Derevienko's team had been excavating Denisova cave for the better part of 3 decades. They knew the stratigraphy intimately. They had protocols and systems and long-running arguments about what specific layers represented and how to interpret ambiguous finds. They had built, through decades of careful work, a picture, incomplete, contested in places, but real, of who had used this cave and when. They did not yet have a name for all of them. That summer, in the sediment of layer 11, fragments of a small bone came to light. The distal phalanx, the tip segment of a finger bone, is one of the smallest structural bones in the body. In an adult, a few centimeters long. In a child or young adolescent, smaller still. What came out of layer 11 that summer was fragmentary, several tiny pieces that required real attention to identify as hominin at all. The cave's abundance of cave-bear skeletal material meant that small mammal bones of any kind were common finds, and fragments this size did not immediately declare themselves. Careful assessment confirmed the morphology. The bone was hominin. Its layer made the identification seem straightforward. A hominin in layer 11 at Denisova cave, by the prevailing understanding of the site, was either a Neanderthal, the Altai was within Neanderthal range, and the site had already produced Neanderthal material, or possibly an early modern human. Nobody reached for any other hypothesis. The bone was catalogued, photographed, bagged with its precise find location documented, and integrated into the season's records alongside everything else. Eventually, it was shipped west, to a laboratory in Leipzig, Germany. The scientists waiting for it had spent more than 20 years building the techniques that would be needed to read it. That story begins not in Siberia, not in the Altai, but in a university storeroom in Uppsala, Sweden, in the early 1980s, where a young man with an unusual double education, Egyptology as an undergraduate, molecular biology as a doctoral student, was conducting experiments that his supervisor didn't know about. Svante Pääbo had become obsessed with a question most of his colleagues considered either impossible or not worth pursuing. Could you extract and sequence DNA from an ancient Egyptian mummy? The idea carried its own obvious absurdity. DNA degrades. The body's own enzymes begin breaking it down within hours of death. Bacteria attack it from without. Heat accelerates the process dramatically. But the hot, dry conditions of Egyptian tombs, it turned out, were not the worst possible environment for organic preservation. The desiccation slowed certain forms of decay, and some mummified tissues had retained traces of original biological material for thousands of years. Pääbo's experiments produced results. His findings, published in Nature in 1985, announced to the field that molecular sequences could be recovered from tissues millennia old. The response combined excitement with the kind of careful skepticism good science requires. If this was real, the implications were obviously enormous. If you could read the genetics of ancient Egyptians, what else could you read? But the contamination problem was immediately apparent to anyone who thought hard about the methodology. Ancient biological samples are not sterile. They carry microbial DNA. They carry the DNA of every researcher who has ever handled them, every conservator, every curator. They sit in museum drawers alongside other specimens for decades, exchanging organic material through the slow work of time. Distinguishing a genuine ancient sequence from a modern human contaminant was, in the early years of the field, essentially impossible. And if you couldn't distinguish them, you couldn't trust anything you found. That problem would define the next phase of Pääbo's career, and the career of everyone who followed him into the field. The contamination problem was not just a methodological inconvenience. It was, for more than a decade, an existential threat to the entire enterprise of ancient human genetics. Modern human DNA is everywhere, on gloves, in air, in the water-use clean equipment, on the surfaces of every laboratory where humans work. And PCR, the technique that amplifies tiny amounts of DNA into readable quantities, does not discriminate between ancient sequence and modern contamination. It amplifies everything. A single modern human cell introduced into a sample does not contaminate the result. It becomes a result. The implications were severe. By the late 1980s and into the early 1990s, the ancient DNA field was producing claims that would not survive scrutiny. DNA recovered from fossils tens of millions of years old, sequences that looked implausibly similar to modern organisms, results that could not be replicated between independent laboratories. The field was generating artifacts and publishing them. Retractions followed. Reputations were complicated. The entire enterprise came to be viewed with deep skepticism by mainstream biology, a place where wishful thinking could masquerade as science, and where the glamour of the question was distorting the quality of the answers. Pääbo had his own reckoning with this. His early Egyptian mummy results, re-examined with sharper methodology, did not hold up as cleanly as the original publication had suggested. He was not unique in this. Nearly every early practitioner of ancient DNA work eventually hit the same wall, but the experience settled something in him. Rather than continuing to produce results he couldn't fully trust, he backed up. He spent years working on the controls, the dedicated clean rooms, the authentication criteria, the contamination detection protocols, the requirement for independent replication, the meticulous documentation of every step in a process so prone to invisible error. He was building a methodology for a field that barely existed in reliable form. By the mid-1990s, Pääbo, now at the University of Munich, where he had relocated his laboratory in 1990, had developed protocols rigorous enough to attempt something genuinely consequential. The mitochondrial DNA of a Neanderthal. The specimen was the original, the type fossil from a Neander Valley in Germany, unearthed in 1856, still held at that point in a Bonn collection. From a few milligrams of bone powder, Pääbo's team extracted and amplified a short stretch of mitochondrial sequence. The results, published in Cell in 1997, were unambiguous. The Neanderthal sequence was clearly and significantly different from any modern human sequence, different in a way consistent with the expected divergence from a lineage that had separated from a modern human line several 100,000 years ago, and different in ways that no pattern of modern contamination could explain. The authentication held. The field exhaled. It could be done, but the ceiling was still low. Mitochondrial DNA, present in cells in hundreds or thousands of copies per cell, compared to the 2 copies of nuclear DNA, was recoverable precisely because it was abundant. Nuclear DNA, which contains almost all of a species' genetic information, and where the most interesting biological questions live, was present in ancient bone in quantities so small and in fragments so chemically degraded that the existing sequencing technology simply could not reach it. Then, roughly between 2005 and 2007, the technology changed completely. Next-generation sequencing, massively parallel sequencing, in the technical language, was developed primarily for clinical genomics. It could read millions of short DNA fragments simultaneously, assembling a complete sequence computationally from their overlapping pieces. Its relevance to ancient DNA was immediately apparent to anyone paying close attention. Ancient DNA is naturally fragmented. Years of chemical decay break the double helix into short pieces, typically between 50 and 200 base pairs long. This had been a fatal limitation under traditional sequencing methods, which required longer intact stretches to work. Under massively parallel sequencing, short fragments were not a problem. They were, in fact, the native input format of the method. By 2006, Pääbo, now at the Max Planck Institute for Evolutionary Anthropology in Leipzig, where he had been since 1997, directing the Department of Evolutionary Genetics, was training next-generation sequencing on the Neanderthal genome. The project ran across multiple specimens, multiple laboratories, and several years of sustained effort. The ancient DNA damage signatures, the specific chemical errors that accumulate at the ends of fragments over thousands of years, now serve not as noise to be filtered out, but as authentication markers, proof that the sequences being read were genuinely ancient and not introduced by contamination at some stage of processing. The noise had become a signal. In May 2010, the Neanderthal genome paper appeared in Science. It confirmed the phylogenetic relationship between Neanderthals and modern humans, and it added something unexpected. People of non-African ancestry carry, on average, between 1 and 4% Neanderthal DNA. Interbreeding had occurred. The two species had met somewhere in the ancient Middle East or Western Asia as modern humans first pushed out of Africa, and the encounter had been more than transient. The contact had left a measurable, permanent imprint on the modern human gene pool. The field was still absorbing this when the second paper arrived. The Denisova phalanx had been sampled in Leipzig some months before the Neanderthal paper published. Mitochondrial DNA came back first, as it always does. And what came back was not Neanderthal. It was not modern human either. The sequence diverged from both Neanderthal and modern human mitochondrial DNA at a depth suggesting a lineage that had branched from their common ancestor approximately 1 million years ago, roughly twice as deep as the split between Neanderthals and modern humans. Not a regional variant, not an isolated population, something that had been on a separate evolutionary trajectory for a very long time. The paper appeared in Nature in March 2010. Its title deliberately restrained. The complete mitochondrial DNA genome of an unknown hominin from southern Siberia. Not a species named, not a classification claimed, just unknown. The authors, Johannes Krauss and Svante Pääbo among them, noted carefully that mitochondrial history is not always a reliable guide to full biological relationships, and that nuclear DNA would be needed to say more. But the word unknown in the title of a paper about a hominin bone from a well-studied region of Siberia was not a word the field had expected to see. It meant what it said. They had found something that did not match anything in the existing catalog of human relatives. The field went quiet the way a room goes quiet when something happens that no one has prepared response for. And then they went back to the bone. The nuclear DNA analysis would take the rest of 2010. When it came back, it did not simplify the story. The nuclear genome changed the picture entirely, and it did so in two directions at once. On one hand, it resolved the deepest uncertainty the mitochondrial paper had raised. The nuclear DNA did not place the Denisovans as a million-year-old lineage, cleanly separate from both modern humans and Neanderthals. Instead, it placed them as a sister group to the Neanderthals specifically, a population that had diverged from a Neanderthal lineage roughly 400,000 years ago, after Neanderthals and modern humans had already separated. The Denisovans were not some ancient holdover from a much earlier chapter of human evolution. They were, biologically speaking, Neanderthal cousins, closely related enough to share a distinct clade, separated enough to have spent 400,000 years becoming their own thing. Why, then, did the mitochondrial DNA suggest something so much more ancient? The leading explanation among specialists, not definitively proven, but supported by a growing body of indirect evidence, is that the Denisovans, at some point in their history, interbred with a deeply divergent hominin population that had been in Asia far longer than they had. Mitochondrial DNA is inherited exclusively through the maternal line, and it is possible for a foreign mitochondrial lineage to sweep through a population and replace the existing one without much affecting the nuclear genome, particularly if that population passed through a small bottleneck. If a Denisovan group encountered an ancient hominin survivor, some late-persisting representative of a lineage that had been in Asia for 1 million years or more, perhaps a descendant of earlier Homo erectus waves or some other archaic presence, and if the offspring of that encounter contributed daughters to the subsequent Denisovan population, the mitochondrial signature of that encounter could propagate forward indefinitely. In other words, before Denisovans ever met Neanderthals, and long before they met modern humans, they may already have absorbed an even older lineage into themselves. Their genome, from the very beginning, was a record of encounters, a palimpsest, the ghost of a ghost. On the other hand, the nuclear genome opened an entirely new puzzle. It revealed that modern human populations carried Denisovan ancestry, but not the populations you would expect. The cave is in Siberia. If Denisovans had interbred with modern humans in the Altai region, you would expect the highest Denisovan signal to appear in the populations geographically closest to cave. The people of Siberia, Central Asia, the broad interior of the continent. That is not what the data showed. The populations with the strongest Denisovan signal in their genomes were not in Siberia. They were in Melanesia, in Papua New Guinea and the surrounding islands, and in Aboriginal Australians, and in indigenous populations of the Philippines, thousands of kilometers from the Altai, in some of the warmest, most tropical environments on Earth, in populations whose ancestors had never been anywhere near a limestone cave in the mountains of southern Russia. The December 2010 paper, authored by a team led by David Reich at Harvard and Svante Pääbo in Leipzig, estimated the Denisovan ancestry in Melanesian genomes at approximately 4 to 6%. Not a trace, not a residual signal at the edge of statistical detection. A substantial fraction of the genome, comparable to the Neanderthal signal in non-African modern humans, inherited from a population that the physical evidence says lived in Siberia. The implication, unavoidable once you stare at the numbers, was that the cave was not the center of the Denisovan world. The cave might barely have been the edge of it. Whatever the Denisovans were, however far they ranged, however they organized themselves across the extraordinary span of their existence, the single cave in the Altai where their bones happen to preserve well enough to survive is not the answer to where they lived. It is an accident of preservation in a world where almost nothing preserves. The Denisovans were somewhere else. Many places, probably places where the soil was too wet, too acidic, too warm to keep bone intact across the relevant timescales. Places where no one has yet thought to look, or looked without finding, or found without recognizing what they had. The numbers in the December 2010 paper were pointing toward a vast, invisible empire, a people spread across the breadth of the largest continent on Earth, present for hundreds of thousands of years, and known from the bones of perhaps 5 individuals. It was the beginning of the field, and it was already clear that the field had an enormous amount left to find. But there was something else in the sediment of Denisova cave, something that nobody had yet looked for carefully enough, something that would, 8 years later, turn an already remarkable story into one of the most extraordinary finds in the history of human paleontology. The bone fragment that would eventually be designated Denisova 11 was, on first inspection, not remarkable in any way that anyone could see. It was a small piece of cortical bone from a long bone, the dense outer shell that forms the structural wall of limbs like the femur or the humerus, roughly 2.5 centimeters at its longest dimension. Its surface had the smooth, slightly yellow quality of ancient bone, preserved in cool conditions. Under normal circumstances, a fragment this size and this undiagnostic, no distinctive morphology, nothing to place it as one element of the skeleton rather than another, would be cataloged and set aside. There are thousands of such fragments in the Denisova cave collections. Most yield only the most basic information, that something lived and died and left pieces of itself in the sediment. Beyond that, silence. What changed this particular fragment's fate was not anything anyone noticed in the cave. It was a technology developed for a different purpose entirely. Zooarchaeology by mass spectrometry, zooms, is a technique for identifying the biological origin of bone fragments through their collagen protein signatures. Every vertebrate species produces collagen with minor, but consistent differences in amino acid sequence. A mass spectrometer can read those differences from just a few milligrams of bone powder, generating a protein fingerprint that can be matched against a reference library. The technique is far faster and cheaper than DNA analysis. It requires only a tiny sample, and it can process large numbers of fragments in parallel. Beginning around 2017, researchers in Pääbo's group and collaborating teams began running unidentified Denisova cave bone fragments through zoom screening. The logic was straightforward. If you can identify which fragments are hominin before committing to the costly process of DNA extraction, you can find needles in haystacks without destroying the haystack in the search. Among the many thousands of fragments screened, a small number produced collagen signatures consistent with human relatives rather than cave bear or deer or horse. Denisova 11 was one of them. The DNA extraction confirmed the collagen result, and the mitochondrial genome that came back first, as it always does, because mitochondrial DNA survives better in ancient bone than nuclear DNA, returned to sequence the Leipzig laboratory now had precedent for Neanderthal. The mitochondrial lineage placed this individual among Western European Neanderthal populations, more closely related to the specimens from Vindija Cave in Croatia than any other reference group in the database. All right, A Neanderthal in Denisova cave. The cave had already produced Neanderthal material. Nearby Shagarskaya cave had Neanderthals. A Neanderthal designation here was a confirmed possibility, not a surprise. Then the nuclear genome came back. Roughly half of the nuclear DNA, the full pair of chromosomes that carry almost all of a species' genetic information, inherited one complete set from each parent, was Neanderthal, and roughly half was Denisovan. The fractions held up across every analytical method the team applied. This was not a statistical trace from ancient population mixing in some distant ancestor. This was not the 4 to 6% Denisovan signal in Melanesian genomes, or the 1 to 4% of Neanderthal ancestry in living Eurasians. This was 50%. Her chromosomes were, in the most literal sense, half one thing and half the other. Her mother, contributing one complete set, had been a Neanderthal. Her father, contributing the other, had been a Denisovan. She was a first-generation hybrid. Her parents had met, and their meeting had produced a child who died young. The bone's cortical thickness, analyzed against reference collections of known-age individuals, indicated she was at least 13 years old at the time, probably in early adolescence, perhaps older. Beyond that, the bone does not speak. Not her face, not her language, not her name, not whether she grew up among her mother's people or her father's or in some community that held both. Her consciousness, what she saw and thought and feared, is gone as completely as anything can be gone. The paper appeared in Nature in August 2018, authored by Svante Pääbo and more than a dozen co-authors. The nickname Denny emerged in media coverage and stuck. The scientific community received the finding with something close to vertiginous wonder, not because first-generation hybrids are theoretically surprising, but because the probability of recovering one from a fossil record seemed vanishingly small. Of all the individuals who ever lived in or near this cave, of all those whose bones might have been preserved in its sediment and survived into the modern era and been collected and stored, this fragment happened to be from an adolescent girl whose parents belonged to two different species. Or, stated differently, if one first-generation hybrid existed in the record, how many others had lived and left no recoverable trace at all? The paper, though, did not stop with the confirmation of hybrid status. It kept going into the father's genome, and what it found there added another layer to a story that was already almost incomprehensibly strange. The Denisovan father, reconstructed from Denny's paternal chromosomes by computational separation of the two parental contributions, was not genetically simple. His own nuclear genome carried evidence of Neanderthal ancestry, Neanderthal sequences embedded in a predominantly Denisovan genetic background, present at a level consistent with an interbreeding event sometime in his own ancestry or in a recent history of his population. This was not the same Neanderthal contribution that Denny had received from her mother. It was older, more fragmented, more diluted by recombination across generations. The signature of a mixing event that predated Denny's parents' own meeting by some indeterminate span. What this means is that the encounter that produced Denny was not a collision between two populations that had been genetically isolated from each other, since their lineages first diverged 400,000 years earlier. Her Denisovan father's people had already encountered Neanderthals at some prior point. The two groups had mixed before, possibly more than once. What looks from a distance like a singular event, two species meeting in a Siberian mountain cave, resolves up close into something more like a long history of contact, a relationship, however intermittent, however asymmetric, however difficult to characterize from this distance. The picture this paints of Pleistocene human life is not the model that dominated the field for most of the 20th century. The older framework described these populations as bounded, isolated, deeply separate, essentially different peoples living in essentially parallel worlds, making occasional violent contact that left little lasting mark. That model was already under pressure from the basic fact of Neanderthal and Denisovan interbreeding. Denny's genome, and what it reveals about her father's ancestry, presses harder still. These were not hermetically sealed peoples. Whatever walls existed between them had gaps. The genetics tells us the contact was sustained enough, repeated enough, distributed enough geographically to produce children who survived, contributed to subsequent populations, and whose descendants carried a record of those meetings into the present. What the genetics cannot tell us is what any of it felt like from the inside, whether those meetings were sought or accidental, cooperative or violent, remembered or forgotten, whether Denny's community considered her ordinary or unusual, whether she herself knew she was different. We have her genome and a fragment of her bone. The rest is silence. But the implication holds regardless of what we cannot recover. If Denisovans and Neanderthals were meeting and producing viable offspring, the question that follows is unavoidable. What happened when modern Homo sapiens arrived at the edge of this world? The path that brought modern humans to the edge of Denisovan territory was long, and it ran through some of the most geographically varied terrain on Earth. The dominant model, refined and revised over the last 2 decades by a combination of archaeological finds and genetic analysis, places a main dispersal of modern Homo sapiens out of Africa sometime after 70,000 years ago, with the most substantial wave pushing east through the southern coastal corridor of Asia, perhaps 60 to 50,000 years ago. These were fully modern people, anatomically indistinguishable from any person alive today, carrying a full behavioral repertoire that the archaeological record of the period attests: composite tools, personal ornamentation, long-distance exchange networks, art. Moving fast by evolutionary standards. Highly adaptable, pushing into environments that no previous hominin had occupied in quite the same way. By the time they were deep into South Asia, they were entering territory the Denisovans had known for hundreds of thousands of years. What happened next is the central empirical question of Denisovan population genetics, and the honest answer is that we are still in the early stages of working it out. What we know for certain is the outcome. Modern human populations today carry Denisovan ancestry, and the signal is not uniformly distributed. It is concentrated in specific places, in specific populations, in ways that reflect where and when encounters happened. The picture that has emerged from a decade of comparative genomics is, in broad outline, this. There were at least 2 distinct interbreeding events between modern humans and Denisovans, involving 2 genetically different Denisovan populations. The first affected the ancestors of populations now living in mainland East Asia and parts of mainland Southeast Asia. The second, producing a stronger signal at higher levels of ancestry, affected the ancestors of Melanesian populations, Aboriginal Australians, and certain indigenous groups in the Philippines. These 2 Denisovan populations were themselves genetically distinct from each other, not different ethnic groups within a continuous species, but populations separated by enough time and distance that researchers have described them as being at least as different from each other as Neanderthals from different parts of Europe. 2 Denisovan worlds, overlapping at their edges, each eventually encountering modern humans in a different place and at a different time. The population responsible for the higher ancestry in Melanesian and Australian genomes, the southern Denisovans, as they're often called, though the name is geographic inference rather than established fact, appears to have been the more divergent of the 2. Their genetic distance from the Siberian Denisovans suggests a separation that goes back, by some estimates, to several 100,000 years ago. 2 branches of the same species, or perhaps 2 species related closely enough to be lumped under the same name for convenience, living in different corners of an enormous continent and developing differently across the long Pleistocene. The ancestors of Melanesians and Aboriginal Australians carried Denisovan ancestry in the range of 4 to 6%. In some groups, among the Ada Magbukan of the Philippines, in research published in 2021, levels of Denisovan ancestry reach 5% or higher, putting them among the most Denisovan admixed populations yet identified anywhere. These are not traces at the edge of detection. They are a substantial contribution to the genomes of millions of living people, inherited from encounters that happened somewhere in the island chains of Southeast Asia, perhaps 50,000 years ago or more. Somewhere, that qualification matters. The precise geography of the interbreeding events is not known. The ancestral populations of Melanesians and Australians crossed open water at some point. The deep oceanic trenches of Wallacea, the island chain that lies between the shallow Sunda Shelf and the Sewell Shelf on which Australia and New Guinea sit, remained as open sea even at glacial sea level lows. No land bridge existed. The ancestors of Aboriginal Australians and Melanesians made a genuine ocean crossing, one of the earliest maritime ventures in human history. requiring watercraft of some kind, and a willingness to sail past a visible horizon into unknown water. Whether the interbreeding happened before that crossing, somewhere in the island chains approaching Wallacea, or after it, on the Sewell side of the oceanic gap, the genetics does not say with precision. What the genetics says is that it happened, and that the population they encountered was one whose evolutionary history extended back through the entire duration of the Pleistocene in this part of the world. The two introgression events implied by the comparative genomics are not the same event repeated. The genetics is clear on this. The Denisovan ancestry in mainland East Asian populations comes from a source genetically closer to the Siberian Denisovans whose bones we found. The ancestry in Melanesian and Australian genomes comes from a source more divergent, a population that had been separate from the northern Denisovans long enough to have drifted substantially in genetic character. Two meetings, two populations, two different chapters of the same long story. What the interbreeding events tell us about the nature of the encounters is inevitably limited. Genetics can detect that mixing happened. It cannot recover intention, context, or experience. What it can tell us is something important about the aftermath. The offspring of these encounters were fertile. Their children survived and had children of their own, and those children had children, and so on, for 50,000 years and more, until their descendants are alive today carrying the evidence of those meetings in their cells. Fertility across what we call species boundaries is not guaranteed. It doesn't work for most of the mammalian taxa ever classified as separate species. The fact that it worked here multiple times speaks to a biological proximity that the formal species distinction can obscure. Neanderthals, Denisovans, and modern Homo sapiens were different enough in morphology and evolutionary history to be distinguished. They were similar enough in the fundamentals of reproductive biology to have children. The scale of the ancestry also speaks to something. For the 6% is not the product of a single encounter between two individuals who happened to cross paths. To reach that level of representation in the modern gene pool and to maintain it across 50,000 years of subsequent population history requires either repeated contact, a large founding population of hybrids, or both. The meetings were not isolated incidents. They were part of something more sustained. What exactly that something was, we cannot say. Whether these were peaceful exchanges at shared resource sites, seasonal overlaps in territory, long-term cohabitation of the same landscapes, or something more complicated than any of those framings allows. The genetics is silent on all of it. What it is not silent on is the product. The descendants of those encounters are alive in substantial numbers. On nearly every island from the Philippines to Fiji and beyond, the Denisovans gave something to these people that has been carried forward through 50,000 years of every change the world has thrown at those populations. The question is what, precisely, they gave. Not just sequences of DNA, not just a percentage on a population genetics chart. What, in biological terms, does Denisovan ancestry actually do? The paper that answered that question arrived in Nature in July 2014, and it arrived as a near-perfect scientific story. A mystery, a method, and a reveal that nobody had quite expected. The mystery was the one stated at the very beginning of this documentary. Tibetans live at altitude. They don't suffer the consequences altitude imposes on everyone else. The specific molecular mechanism involved a variant in EPS1, a gene encoding a protein called HIF-2-alpha, hypoxia-inducible factor 2-alpha, which sits near the top of the cellular signaling cascade governing the body's response to low oxygen. In most humans exposed to chronic altitude, the HIF pathway upregulates erythropoietin, driving the bone marrow to produce more red blood cells. The Tibetan variant appears to dampen this response, to partially uncouple the low oxygen signal from the red blood cell overproduction that normally follows it, allowing cells to adapt their oxygen extraction efficiency without flooding the blood with extra hemoglobin. The blood stays thin. The heart stays healthy. The body functions. What made the EPAS1 story remarkable was not the gene itself. It was the haplotype. A haplotype is the extended pattern of genetic variation across a region of the genome. Not just a single mutation, but the entire surrounding landscape of variants that tend to travel together because they sit close to each other on the chromosome and are rarely separated by recombination. You can trace the ancestry of a haplotype independently of the functional variant it carries, because the surrounding pattern reveals where it came from even when the variant itself is too recent or too simple to speak. The EPAS1 haplotype in Tibetans didn't look like anything in the Han Chinese genome. Despite Tibetans and Han Chinese sharing a recent common ancestral population, it didn't look like anything in any other modern human population database. It looked, with a clarity that left the authors of the 2014 paper careful but unambiguous in their language, like the Denisovan genome sequence from Denisova cave. Emilia Huerta Sanchez at the University of California, Berkeley, and her colleagues published the finding under a title that stated the conclusion plainly: altitude adaptation in Tibetans caused by introgression of Denisovan-like DNA. The Tibetan EPAS1 haplotype was not the product of mutation and selection within the modern human gene pool over the last 30,000 years. It was inherited already formed from a Denisovan population that had been living at altitude or in altitude-adjacent environments long enough to develop and fix this variant through natural selection in their own population. Then modern humans arrived. They encountered the Denisovans. They interbred. Some of the offspring carried the EPAS1 variant. At low altitudes, the variant is essentially neutral. It doesn't hurt you, but it doesn't especially help you either, which is why it never spread through global human populations in the 50,000 years since those encounters. But when the descendants of those meetings moved onto the Tibetan Plateau, the variant became intensely advantageous. Natural selection, operating over the roughly 30,000 years that modern humans have been on the plateau, drove its frequency from low to overwhelming. Today, approximately 87% of Tibetans carry it. In Han Chinese, who share a common ancestral population with Tibetans but live overwhelmingly at low altitude, the frequency is in the single digits. The selection signal in Tibetans for this variant is one of the strongest signatures of recent natural selection detected anywhere in the human genome. Powerful selection, acting fast, on a variant that modern humans could not have developed independently in the time available. They didn't develop it. They inherited it from a people who had already done the work. This is the callback to the opening of this story. The person on the Tibetan Plateau, breathing without distress, living without the pathologies that altitude forces on every other human body. They are doing this, in part, because somewhere between 40 and 60,000 years ago, one of their ancestors met a Denisovan. That meeting produced a child. That child had children, and in one of those children, by the ordinary accident of recombination, the EPAS1 haplotype happened to be on the chromosomes that were passed forward rather than the ones that weren't. Eventually, a human being stood on the Tibetan Plateau carrying the haplotype, was healthier for it than their neighbors, had more surviving children, and here we are. There is no direct line. There is no narrative of intention. Nobody gave anyone a gift. It is just what happens when populations meet. Genetic variation flows between them, and some of that variation turns out to be useful in ways that only become apparent later, in environments that didn't exist at the time of the encounter. But it is real. It is happening now, in the bodies of millions of people, and it came from the Denisovans. EPAS1 is the clearest case, but it is not the only one. In the immune system, the picture is comparably striking. Among the most variable regions of the human genome are the HLA genes, human leukocyte antigen genes, which encode proteins that sit on the surface of cells and help the immune system distinguish self from non-self, healthy tissue from infected tissue, ordinary cell from cell harboring a pathogen. HLA diversity is directly tied to immune breadth. The wider the variety of HLA variants in a population, the wider the range of pathogens those individuals can recognize and mount response against. It is one of the most intensely pathogen-driven regions of the genome, under stronger selection than almost anything else in the human genetic landscape. In 2011, a team led by Laurent Abiracht and Peter Parham at Stanford University published a paper in Science examining HLA alleles in modern human populations with high Neanderthal or Denisovan ancestry. What they found was that specific HLA alleles in modern Asian and Melanesian populations appear to be Denisovan-derived. Sequences absent or very rare in African populations, who carry no detectable Denisovan ancestry, but common in populations with established Denisovan admixture, and most similar in exact sequence to the Denisovan genome. HLA*11, which is among the most common HLA alleles in many East Asian and Southeast Asian populations today, carries signatures consistent with Denisovan origin. This is not a marginal variant. It is one of the dominant immune response profiles in some of the world's most populous regions. If its origin is indeed Denisovan, and the evidence, while not as clean as the EPS1 case, is substantial, then the immune systems of billions of people living today were shaped in part by a population that had lived in Asia for hundreds of thousands of years before modern humans arrived from Africa. The Denisovans have been exposed for all that time to the pathogens of their specific environments, the parasites, bacteria, and viruses of Asian forests and mountains and river valleys. Their HLA variants were tuned to those pathogens in ways no recently arrived African populations' HLA variants could match. Inheriting those variants gave modern humans moving into Asia something they couldn't have generated themselves in the time available, an immune system that already knew some of the local threats. Researchers have identified other regions of the genome where Denisovan-derived sequences appear to be under positive selection in modern populations, where the ancestry is not evenly distributed, as neutral inheritance would produce, but concentrated in ways suggesting the variant is beneficial and has been increasing in frequency. The functional interpretation of many of these regions is still being worked out. The human genome is large. The Denisovan contribution was a fraction of it, and distinguishing true adaptive introgression from statistical noise requires careful analysis across large sample sizes. The field is active and the list of confirmed cases is not long, but it is growing with each new dataset. What has already been confirmed is enough to establish the principle. The Denisovans did not simply disappear. They were absorbed and the material they contributed dispersed into the modern human populations that encountered them, and those populations carried it forward. And natural selection operated on it over subsequent millennia in the same way it operates on everything else in the genome, preserving what worked in context, letting the rest persist as background. The Denisovans are extinct as a biological lineage. There are no people living today who are Denisovans in any continuous, meaningful sense. But there are people living today whose lungs work better at altitude, whose immune systems recognize a wider range of pathogens, possibly whose bodies manage energy differently in cold or humid environments, because of who the Denisovans were and what they had spent hundreds of thousands of years becoming. That is a legacy, an impersonal one, driven by mechanism rather than intention, but real, ongoing, and in some cases a difference between surviving and not surviving. A species that left almost no bones, no tools we can confidently identify as theirs, no art, no cultural memory of any kind. And yet, in the most practical biological terms, they are still here. The mystery then is not whether the Denisovans mattered. The mystery is how little we actually know about who they were. Consider what we know. We know the Denisovans were genetically distinct from Neanderthals and from modern humans, but more closely related to Neanderthals and to us. We know they split from a Neanderthal lineage roughly 400,000 years ago. We know their genome shows signs of a once large, once diverse population spread across an enormous territory. We know they interbred with Neanderthals, producing at least one individual whose bones we have recovered. We know they interbred with modern humans at least twice, in at least two geographically distinct events. We know that their DNA survives in hundreds of millions of living people, and that some of that DNA is performing specific, identifiable biological functions in the bodies it inhabits today. We know that a gene variant they carried, tuned by hundreds of thousands of years of high-altitude life, was passed to modern humans who eventually colonized the Tibetan Plateau, and that this gift is, in the most literal physiological sense, keeping those people alive. We know all of this from a fragment of finger bone, 3 teeth, a sliver of skull, and 1 jaw. The disproportion is almost philosophically uncomfortable. In no other domain of human inquiry would this ratio be considered acceptable, an inference so rich, resting on a foundation so thin. And yet the science is sound. The methods have been checked and replicated and refined. The genome does not lie in a way that a misidentified fossil can lie, or a mistranslated inscription, or a biased historical account. The chemistry is either there or it isn't. The sequences either match or they diverge, and the conclusions drawn from them carry the weight of hard, verifiable work. The problem is not the quality of what we know. The problem is the quantity of what we don't. We have no Denisovan skeleton. We cannot say, with anything approaching confidence, what a Denisovan looked like in full. We have no confirmed Denisovan tools, no assemblage of stone or bone that a researcher can point to and say, these people made this. And we know it because we connected the artifacts directly to identified individuals through secure stratigraphy and confirmed genetics. We have no Denisovan art, no evidence of symbolic marking, no pigment use attributable to them, no deliberate burial. We have a polished stone bracelet that may be theirs and may not be. We have layers of sediment full of animal bones bearing cut marks, and tools made with real sophistication and charcoal from ancient fires, but nothing that can be hung on the Denisovan name with the evidentiary security the field requires. A species that may have occupied more territory for more time than any other archaic human lineage, and their cultural record is, so far, a blank. Then, in 1980, a Tibetan Buddhist monk found something in a cave. Baishiakar's cave sits near the town of Shaha, in Gansu province on the northeastern edge of the Tibetan Plateau. The elevation is approximately 3,280 meters, over 10,000 feet above sea level, high enough that most of the year the winds are sharp and the air thin. The cave opens in a pale limestone cliff above a valley carved by the Yellow River's upper tributaries. The surrounding landscape, a mix of alpine meadow and exposed rock typical of the plateau's lower margins. The monk entered the cave, as monks had presumably done for generations, and found a partial lower jaw on the floor or the surface of the deposits. He couldn't have known what it was. He brought it out into the light, carried it down from the cave, and eventually donated it to the sixth Gungtang living Buddha, a revered religious figure in the local Tibetan Buddhist tradition. The living Buddha in turn donated the mandible to Lanzhou University, where it entered a collection and for decades sat largely unstudied. The problem was partly classification and partly timing. The mandible was clearly archaic. Its morphology didn't match modern human anatomy, but the tools to say precisely what didn't yet exist in a form anyone could apply without DNA. And no DNA survived in the bone. The plateau's freeze-thaw cycles, the altitude's particular combination of UV radiation, temperature fluctuation, and aridity, had degraded the ancient DNA in the specimen beyond any usable recovery. By the late 2010s, the bone had been identified as potentially significant, but remained unconfirmed. What changed was the proteins. Collagen, the structural protein that forms a matrix of bone, degrades more slowly than DNA, far more slowly, under the right preservation conditions. Collagen peptides can survive in bone for millions of years, readable by mass spectrometry through their characteristic fragmentation patterns. Crucially, collagen sequences differ between species in ways that reflect the underlying DNA differences. A Denisovan, a Neanderthal, and a modern human would produce collagen with subtly different amino acid sequences at key positions, and those differences are preserved even when the DNA itself is entirely gone. A team led by Fao Chun at the Institute of Tibetan Plateau Research and Jean-Jacques Hublin at the Max Planck Institute for Evolutionary Anthropology retrieved the Xia mandible for fresh analysis. They extracted collagen from the bone and ran the peptide sequences against reference data. The results, published in Nature in May 2019, were unambiguous. The collagen matched the sequence expected for a Denisovan, based on the genetic differences visible in the Denisova cave genome. Uranium series dating of a carbonate crust encasing part of the specimen placed its minimum age at 160,000 years. The bone itself could be older still. The Denisovans had been on the Tibetan Plateau 160,000 years ago, an altitude of over 3,000 meters, in a precise high-altitude environment where the selection pressure for the EPS1 gene variant would have been strongest. There they were, not an inference from gene distributions in living populations, not a prediction from a model, physical evidence, pinned to a place and a time. The cold open of this documentary asked why the people of Tibet can breathe where others cannot. By the time you reach the 2019 paper, the geography of the answer is complete. The Denisovans were on that plateau. They were there long enough to develop a precise, efficient adaptation to low oxygen conditions. When modern humans arrived in Asia and encountered Denisovans somewhere along the route to the plateau, some portion of that adaptation transferred. Their descendants eventually reached the plateau itself, and the variant did exactly what had been shaped by hundreds of thousands of years of selection pressure to do. There is more. In 2021, a team applying the sediment DNA technique to material from Baishia Karst Cave, the same cave where the monk found the jaw, extracted Denisovan DNA from the deposits without any bone at all. The sequences appeared in layers dated after 100,000 years ago. Some appeared in layers that may be as recent as 45 to 60,000 years ago. The implication is extraordinary. Denisovans were present in this cave on the Tibetan Plateau during a period that broadly overlaps with the arrival of modern humans in Central and East Asia. They may have still been there or recently gone when the ancestors of the people who now carry that EPS1 variant were moving through the region. The meeting may have been close in time, as well as decisive in consequence. The year after the Shah paper appeared, a different discovery announced itself from a completely different direction and complicated the picture in ways the field is still working through. In the summer of 2018, an elderly man named Yi Changju told researchers at the Hebei Geo University about a skull. His grandfather had found it in 1933 during construction of a bridge over the Songhua River near Harbin in the northeastern Chinese province of Heilongjiang. The workers, according to the family account, recognized it as something extraordinary. The grandfather, apparently wary of the Japanese authorities in occupying Manchuria, who were known to take an active interest in paleontological finds, wrapped the skull and hid it at the bottom of an old well. He kept the secret for the rest of his life. Before he died, he told his family where it was. The family retrieved it and held it privately for decades before Yi Changju finally brought it to scientific attention. What emerged from that well, cleaned and examined in Hebei and then in Beijing, was one of the most remarkable hominin fossils ever recovered from Asia. The skull is massive. The estimated brain volume falls in a range of 1,420 cubic centimeters, within modern human variation at the upper end. But the face is enormous, deep, broad, with huge square eye sockets and a low, flat frontal bone. The brow ridges are thick. The back of the skull is wide and rounded in a way that doesn't fit neatly into the existing catalog of Asian archaics. It is very large, very robust, and very strange, and very old. Uranium series dating has placed it at over 146,000 years, possibly considerably more. In June 2021, 3 papers in the journal The Innovation proposed that the skull represented a new species, Homo longi, colloquially dragon man, from the Chinese name for Heilongjiang province, which means Black Dragon River. The papers further argued, controversially, that Homo longi might be the closest known relative of Homo sapiens, closer to us, phylogenetically, than the Neanderthals are. The field did not receive this quietly. Some researchers broadly agreed with the analysis. Others questioned the phylogenetic methods, arguing that the tree-building approach used in the papers was sensitive to assumptions that hadn't been adequately tested. Some pointed out that the isolation of the skull, no associated fauna, no stratigraphy, no other bones from the same individual, made confident placement anywhere in the hominin family tree extremely difficult. And then there was the Denisovan question. Several researchers, examining the skull's morphology and its geographical and temporal context, raised the possibility that the Harbin skull might represent a Denisovan, or at least a member of the same broader population, whose bones are known from Denisova cave. The dental dimensions, where they could be estimated from the tooth sockets, are consistent with the large molar sizes seen in confirmed Denisovan specimens. The geography is right. The approximate time period is right. The skull is in the right part of the world at roughly the right time to belong to the group whose genome we've already sequenced, if that group ranged across China, as the genetics implies it did. The countervailing problem is that without ancient DNA or ancient proteins from the Harbin skull, no one can say definitively. The bones history, hidden in a well unknown to science for 80 years, means that the preservation conditions are uncertain, the surrounding context irretrievably lost, the stratigraphic relationship to any dated deposit impossible to establish. It is a magnificent object with a damaged provenance, and the debate around it is the honest scientific response to exactly that situation. What the Harbin skull does, regardless of how that debate eventually resolves, is make the Denisovan question more vivid and more urgent. If it is a Denisovan or a close Denisovan relative, we suddenly have a face, partial, inferential, worked out from a skull whose owner died before our own species reached this part of the world. A massive, heavy-browed, large-brained face unlike anything in the familiar portfolio of human types, belonging to a people who range from Siberian mountains to Chinese river valleys to the Tibetan Plateau. If it is something else, a lineage closely related to Denisovans but distinct, or a regional variant of Homo heidelbergensis with its own separate trajectory, then the Middle Pleistocene of Asia was even more populated with human diversity than the Denisovan story alone implies. Neither conclusion diminishes a story. Both expand it. There's a technique that has begun to cut through some of these ambiguities, and it is worth pausing on because it represents the next frontier of Denisovan research. Environmental ancient DNA, EDNA, is genetic material that living organisms shed into their surroundings. In skin cells, in hair, in feces, in urine, in the microscopic detritus of ordinary life that accumulates in soil and sediment over time. In 2017, Matthias Meyer and colleagues at Leipzig published findings in Science demonstrating that ancient hominin DNA could be recovered from cave sediment without any bone at all. The technique was applied to Denisova cave and found Denisovan DNA in sediment layers where no identified bones had been recovered, extending the known Denisovan occupation of the cave further than the physical specimens alone allowed. The implications are significant. If Denisovan DNA can be extracted from sediment at Denisova cave, it can potentially be extracted from sediment at other Asian caves where Denisovans may have lived. Teams are now applying the approach to sites across Central Asia, China, and Southeast Asia, places where the geological conditions might have preserved genetic material even in the absence of intact bone. The search is expanding from the physical to the molecular, from the object you can hold to the chemistry you can only read. It is a slower, more painstaking process than finding a fossil, and its results are less immediately dramatic. But it may ultimately tell us more about where the Denisovans actually were within decades of traditional excavation. What it cannot tell us, what nothing yet recovered can tell us, is what the Denisovans were like as a people. Not what they thought or believed, or made or sang. Not what stories they told, or what they were afraid of in the dark. Not whether they loved their children the way most social mammals love their young, or whether they mourned their dead, or whether they had any concept at all of the distinction between the living and the dead. The genetics can infer something about the color of their skin and hair and eyes, can tell us about their immune systems and their altitude adaptations, can estimate the population sizes of ancient groups, and trace the movements of lineages across continents. But it cannot cross the threshold into consciousness or culture. It is biology, not biography. We are left, therefore, with a paradox that has no clean resolution. The Denisovans are among the best characterized archaic human populations in genomic terms. Their evolutionary relationships mapped, their contributions to modern human biology identified, and in some cases functionally understood, their history of encounters with other human species written in the DNA of millions of living people. And they are, in every other sense, almost completely unknown. Hundreds of thousands of years of human life, of people waking and sleeping, finding food and avoiding predators, raising children, losing companions, navigating the relationships of whatever social group they lived in, and none of it recoverable. Not a word, not an image, not a memory that anyone living can trace back to them. On October 3rd, 2022, the Nobel Assembly at Karolinska Institutet in Stockholm announced that the Nobel Prize in Physiology or Medicine for that year would go to a single recipient, Svante Pääbo, for his discoveries concerning the genomes of extinct hominins and human evolution. It was the first Nobel Prize awarded for work in ancient DNA, and the first for a specific field of paleogenomics, the reading of genomes from the deep past. Pääbo had spent more than 4 decades building the methods and standards and laboratory culture that made the science possible, and the Nobel recognized not just the technical achievement, but the conceptual transformation produced, the understanding that modern humans are not a clean line, but a braided river, that our genome carries the contributions of people we cannot name, that the story of who we are reaches back much further, and runs through many more kinds of human beings than anyone suspected a generation ago. The prize committee said his discoveries had provided insights into what makes us uniquely human, the Denisovans. The discoveries that opened the deepest parts of that question were front and center in the citation. And yet, On the day the prize was announced, the Denisovans were still almost entirely unknown, still essentially invisible, still speaking only through the chemistry of their bones and the fractional genomic traces they left in populations whose ancestors had encountered them tens of thousands of years before. The Nobel Prize was awarded. The cave continued to give up its sediment a careful trowelful at a time. The Asian soil above 100 unexcavated sites continued to hold whatever held. The techniques are improving. The search is widening. There are caves in Laos, in the Philippines, in southern China, in the limestone karst country of mainland Southeast Asia that have barely been touched by systematic excavation. There are plateau margins in Tibet and Yunnan and Sichuan where the geological conditions might be right for preservation. There's a whole continent of possibility. The next bone, if it comes, could change everything we think we know. Again, go back to the plateau. Someone is breathing. The air is thin, and the sky above the plateau is a shade of blue darker than most people ever see. The atmosphere here is thin enough to let more space through, and the cold comes off the surrounding mountains in the afternoon like a second presence in the landscape. The person breathing is not extraordinary, in the sense that they would recognize a farmer, perhaps a monk, a child walking home along a gravel road. They are doing what people do on an ordinary afternoon. Being alive, in a place where being alive demands certain things from the body, giving the body those things without particularly thinking about it. Their blood does not carry the signs of altitude-induced strain. Their hemoglobin is not elevated. Their heart is not enlarged. Somewhere in the cells of that person, not visible, not felt, not present in any way they will be aware without a DNA test and a specific research question, a stretch of the genome does quiet work. A region of the chromosome not far from the gene EPS1, inherited from a parent who inherited it from a parent, going back in an unbroken chain for perhaps 30,000 years, back to an ancestor who lived lower on the slopes, and met someone who lived higher, much higher, for much longer. We know now who that someone was. The Denisovans gave modern humans no gift in any intentional sense. There was no transfer of knowledge, no teaching, no moment in which a Denisovan sat down with an anatomically modern human and explained what they knew about surviving at altitude. What happened was biological and impersonal. Populations encountered each other, had children, and through the ordinary mechanics of inheritance and natural selection, some of what the Denisovans had spent hundreds of thousands of years becoming. Their altitude physiology, their immune system calibrations, possibly other adaptations we're still identifying, was preserved in the bodies of people who carried it forward into a world the Denisovans never knew. This is not unique to them. Neanderthal DNA exists in the genomes of virtually all non-African modern humans, and some of it is demonstrably adaptive, shaping immune responses, influencing biological processes in ways researchers are still mapping. Ancient populations throughout human history have met and merged and diverged, the genetic record of those encounters outlasting every other trace of the people involved. The human genome is not a clean, linear document. It is an archive of meetings. But the Denisovans represent something extreme within that general truth. They are the human population for whom the ratio of genomic knowledge to any other kind of knowledge is most severely lopsided. The species we understand most completely through biology and most poorly through everything else. We can say something about their immune systems, but nothing about their language. We know something about their altitude adaptations, but nothing about their social structures. We have traced their population history across hundreds of thousands of years in the width of the largest continent on Earth, and we cannot name a single individual among them, except the ones we have invented nicknames for, because the alternative was clinical designation in every sentence. We cannot name their peoples or their territories. We cannot describe their seasons of hardship or abundance. We do not know what they called the cave. That is a particular kind of loss, and it is worth sitting with for a moment, not a sentiment. The Denisovans did not know they would be lost, and their loss is no more, no less than the loss of the countless human populations who lived and died before any record was kept. But as an epistemological fact, we are extraordinarily good now at a certain kind of knowledge, the chemistry of ancient identity, the molecular fingerprints of long-ago encounters, the deep structure of biological relationship across populations separated by hundreds of thousands of years. And we remain completely unable to reach another kind of knowledge, what it was like to be a Denisovan on an ordinary afternoon, what the valley looked like from the cave entrance in the season when the river was high, what the stories were that people told each other after dark in the limestone room above the water, with the cold coming in from outside and the fire going in the center. Svante Pääbo, accepting the Nobel Prize in Stockholm in December 2022, described one of the original motivations behind his decades of work: the possibility of recovering the individuality of the dead, of reaching past the generic category of a species into the specific fact of a person. That ambition is, in the Denisovan case, only partly fulfilled. We can distinguish individuals from one another in the cave sediment. We know something about Denny's parentage and her sex and her approximate age at death and the ancestry of both her parents. But the full individuality, the thing that makes a person rather than a specimen, that remains on the far side of what the chemistry can reach. What we have instead is something more durable than any individual story, because it is still running. The consequence of those ancient encounters is alive today, in the Tibetan farmer breathing without strain at 15,000 feet, in the Melanesian fisherman whose immune system carries HLA variants tuned over hundreds of thousands of years, to the pathogens of Asian environments that no African-derived immune landscape had encountered before, in the millions of people across island Southeast Asia and the Pacific who carry, distribute across their chromosomes, a biological inheritance from a people whose cultural world left no recoverable trace. The Denisovans are part of the people who came after them, not metaphorically, not as a poetic turn of phrase, but chemically, physically, functionally. Their biology is inside human beings doing specific things right now, today, and will be inside human beings 100 years from now and 1,000 years from now, as long as the populations carry and survive and continue. That is a kind of immortality, an impersonal one, without memory or intention behind it, but real. The cave in the Altai is still being excavated. Every summer, the team from Novosibirsk returns to the valley of the Anui River. The sediment comes out a careful trowelful at a time, screened and sampled and logged. Cave bare bones continue to appear, as they have always appeared in every layer at every depth. Sometimes something else appears. The sediment goes deep. The lower levels have not been fully explored. There may be more bones, more individuals, more encounters preserved in the cold, and there may be objects whose makers can finally now be identified. Somewhere in Asia, perhaps already in an unstudied collection somewhere, perhaps in a cave in Yunnan or Laos or on the margins of the Tibetan Plateau, there may be the fossil that changes the picture again, a skull with enough morphology to answer the questions the jaw from Shah opened, a skeleton that lets us finally see what the Denisovans look like, standing upright, looking back at us with whatever degree of recognition that kind of eye contact would allow. It may come next year. It may come in a generation. It may already been found and not yet recognized for what it is. The cave entrance is open. The temperature inside is just above zero. The temperature it has always been. The temperature that has kept what it has kept across the whole improbable length of its occupation. Outside, the Anui River runs cold and clear over stones in the late afternoon. The mountains above the valley hold the last of the light for a moment, and then it goes. Somewhere in the sediment below the current floor, the past is still being held in place, waiting, as it always has been, for someone to come and read it.